welcome. So, we are still continuing with manganese. So, today we will see some more examples. So, this manganese we have identified when it is present as an oxo-hydroxo cluster. And in photosystem it is involved in the center, which is responsible for oxygen evolution. And we have also identified there that not only manganese, but we have also calcium. So, we have calcium, we have manganese intimately connected to one calcium, and also we have one more extra manganese, and here we have for completion of the Cuban structure we have 4, and sometime this O and OH is connected to that. So, we are just looking for the evolution of O2. So, it is a completely different structure and the reactivity compared to the well known system what we know as our ferredoxin system. So, in case of ferredoxin molecules, they know they serve the purpose of only electron transfer. So, in this particular case it has some other roles to play, because to get this dioxygen we have to use water, and this water should be a very good ligand for this cluster system. So, if we just think of the basic structure, we just look at the basic structure only. So, you have calcium, and calcium what we have seen last time, that it is also connected to the manganese. So, if we have some connectivity pattern like this, and this calcium is highly distorted in nature, and if we have 2 more manganese at these 2 points, so sometime we call it as a distorted adamantine structure. And this particular site is important, and also this particular site where you have the separate manganese. So, like all other catalysis systems and some other cases, people have still proposed, is not yet settled, that you can have a double bonded oxo structure or a triple bonded oxo structure from this manganese, and this calcium is bound to one single water molecule. So, these are the very important structures related to this cluster, that you have the ligand in your hand, and this ligand as well as some of these form, because we have identified that these groups, that O2 minus or hydroxide groups, they are always present in form of manganese oxo manganese basic unit. So, when we have this oxo hydroxo cluster, sometime we also can get in the manganese oxo center like the vanadilyl, molybdenal and uranyl system. So, if you have a highly oxidized form, if we just manganese oxo in this form, it can be manganese 5. So, what we are basically proposing there, so is the basic mechanism for that is that this oxo center is important for this O2 evolution. So, we can generate some highly electrophilic oxo center. So, generation of that is very important on manganese, and this particular one will go for this water molecule. Then if you have adjacent, so you have this entire cluster arrangement. So, if we have this particular water molecule close to this oxo center, it can go for nucleophilic attack to that oxo center. So, we just allow nucleophilic attack from this water molecule, and we from there we get this O2 molecule as the very basic oxygen evolving center. So, this particular manganese motif, that means this entity. So, everywhere you will find this manganese oxo manganese, manganese oxo manganese. So, this particular one, when we find that, so this we are talking about this calcium with attached with water molecule. So, in some other form you can get this particular basic unit as well. So, water bridging, hydroxide bridging and oxo bridging center. So, whenever we have these two manganese adjacent to it, so here also is not that some isolated manganese center like cytochrome P450 or something. That means, if it is not that it is a porphyrin bound manganese center, mononuclear manganese center, then it is attracting the water molecule. This the cluster arrangement is required. That means, you need some adjacent manganese site which is bound to this manganese, which is the catalytic site, through some oxo, hydroxo or water bridging. So, we will see some more manganese centers. So, where you can put more than one manganese center. So, these manganese have been identified in several other molecules. Some of we already know, and we call them as they are present as cofactor in catalases. The most important catalases we have seen, they are heme proteins, so iron porphyrin unit, but there are some other species which is available for their reactivity like that. Again, the disproportionation of hydrogen peroxide, so which is well known to us from the different E0 values, that it can go for its disproportionation reaction. Because it is thermodynamically favorable. So, if you some reaction thermodynamically favorable and we want to just change the corresponding rate of the reaction. So, rapid reaction requires what we require? We require a 2 electron catalyst. So, once we know that for disproportionation reaction we need a 2 electron catalyst. So, we can have a manganese site, that means you can have a 2 manganese site. So, it is again a dimanganese catalyst. So, the manganese bearing catalysis, so it is not that the iron bearing catalase, that means the heme catalysis. So, manganese bearing catalysis, there are also like that heme proteins, you have the peroxidases also. But the mechanism of action is completely different, and superoxide dismutases. So, catalases, peroxidases and superoxide dismutases, and in all these cases we have manganese. So, in case of, so side by side if we see that in case of hydrogen peroxide we need a 2 electron catalyst which can increase the rate of the reaction. Similarly, for superoxide, so we can have a single electron transfer from the superoxide anion. So, this particular one is very much useful if we can have a distorted trigonal bipyramidal arrangement on a mononuclear manganese, and mononuclear manganese initially can bound to water molecule and then to hydroxo function. So, the corresponding reactivity with the superoxide anion can be taken care of by using a mononuclear center. But in case of this disproportionation of hydrogen peroxide, we definitely need the binuclear one. So, here if we just generate simple for ion based manganese system, that means if we have a manganese 3 plus center, we go for reacting this O2 minus, the superoxide anion is reacting with it, liberating O2, and this center can be reduced to manganese 2 plus. Then in the second stage also we can have the corresponding reaction of O2 minus, but now 2 protons will also react, and reaction of these 2 protons with manganese 2 plus in presence of the superoxide will generate now your hydrogen peroxide. So, the superoxide we break into O2 and H2O2, regenerating the center again as manganese 3 plus. So, at this point we are regenerating the oxidized form of the enzyme. So, we regenerating the oxidized form of the enzyme. So, considering that number of electron transfer, so is the 1 electron catalyst in a stepwise manner, we can consume 2 molecules of superoxide anion to produce oxygen and hydrogen peroxide. But for this disproportionation reaction based on manganese, we should have a corresponding dimanganese system, and this dimanganese all the time like our photosynthesis center, that it can be connected with an oxo bridge and one carboxylate bridge, and this carboxylate bridge is coming from glutamate 66. This has nitrogen coordination from histidine 181, and interestingly some distorted coordination which is not very common for first transition series elements like manganese, that it can go for bidentate carboxylate coordination from another glutamate residue, which is glutamate 148. And the third bridging is from hydroxide function. So, that gives us some idea that that both the 2 manganese sites, this particular part can be close to an octahedral geometry, this can also be close to octahedral geometry, because we are accepting 3 bridging units. So, one particular face of the octahedron is shared between these two manganese sites. So, we have other 3 coordinate sites. This is again like this nitrogen is histidine 69, this oxygen from the carboxy end, but this carboxy end is functioning as a monodentate one, which is glutamate 35, and this is water. This is a more or less symmetrical arrangement. Only thing that you have 2 nitrogen, this acid carboxylate bridging, then oxo hydroxo carboxylate bridging. So, any dimanganese system like this, so we have manganese site and we have 2 such manganese unit, and if both the 2 sides are close to octahedral geometry, because this particular part is highly distorted one, and if we look at the bond distances, this is 2.44, which is quite big, and this is 2.36. It is well above the corresponding manganese oxide distance, because this particular one we get in a very distorted way, and this carboxylate function is little bit weakly bound compared to our this particular bridging carboxylate unit. This is in a short range, which is 2.08 angstrom, and the other one is 2.13. Why we are looking at all these bond distances? Because sometimes we find that these bridging groups, like your simple copper acetate, these 2 bridging groups are not symmetrical. So, asymmetric bridging groups are there, and identification of these carboxy end, even it is true for isolated metal salts. That is, if you are talking about manganese acetate, if you are talking about iron acetate, or if you are talking about some other metal acetate, how these acetate groups are attaching to the metal center. So, here you will find, which is also true for different types of iron enzymes like hemerythrin and ribonucleotide reductase, is that you have one such carboxylate unit, which is glutamate. So, you have 3 such glutamate ligand, one is this, another is this, and another is this one. So, depending upon their positioning within the long protein chain, one is functioning as the bridging unit, another is functioning as a bidentate one, and another is functioning as a monodentate one. Still we have some other possibility because of this charge neutralization from the acetate anion, because these are negatively charged. Sometimes this hydroxo function can also be replaced by some acetate function, where one single oxygen of the acetate group can be used for bridging to manganese site. So, this hydroxy function can be equivalent to that of our acetate function coming from the glutamate residues. So, in almost all these cases, whether we can have 2 different types of these catalases. So, sometimes we find that in these catalases are present. One such example is in lactic acid bacteria, in different lactobacilluses. So, in lactic acid bacteria, they are present and almost in a very low oxygen environment. So, almost oxygen free environment they are surviving. So, it is not that you have water or aqueous medium and you have the dioxygen molecule, and which can give rise to the corresponding oxidation of the manganese site from manganese 2 to manganese 3 and then again 3 to 4. So, if this particular system is such that you can move from manganese 2 to manganese 4, then one such mononuclear manganese was sufficient to get 2 electrons out of that. But in this particular case, like all other binuclear systems, it can go for one single electron transfer from one side and another single electron transfer from the other side. So, if we have the enzyme, the dimanganese enzyme for catalase, which is in the oxidized form, we can consider that both the two manganese sites are in the trivalent state. So, this trivalent manganese can go for that. Then in the reduced form, if both the two centers can be reduced, we can have a manganese 2,2 site, and at the intermediate position, which should be the mixed valent one. So, one manganese site can be oxidized and you get manganese 2,3 compound. So, manganese 2,3, then other one also, that if we are able to oxidize one such manganese site to its +4 oxidation state, we get manganese 3, manganese 4 compound. So, these are the different possibilities, and some of them are only observed because you need 2 electron only for the catalytic site. So, all these things. So, you have the oxidized form, you have the reduced form, and when we consider this is oxidized, and sometimes if this manganese 4 is not so common in other cases, we call them as the superoxidized form. So, the basic unit for this corresponding oxidation reaction, that if we have these triply bridging groups and if two of them, that means this particular one, when one single oxygen atom is utilized, that means when all 3 bridging groups are present, you can have some control over the manganese distance, because this manganese distance is very important, because some other reactions are also taking place during electron transfer. So, briefly we will see how the catalytic cycle is operating. So, initially if we have the manganese site, the most common one, that means the oxidized one, that's a trivalent manganese, both of them are trivalent, and one is bridged by hydroxo and another part is bridged by oxo. So, what happens if you just this particular motif, that means if you consider the corresponding shape as a corresponding diamond shape, if we bring the third bridging unit, the acetate unit, how it goes to bind these manganese sites? Because this all 4 atoms will be in the plane of the paper. So, if you have acetate, it will be above the plane of the paper or below the plane of the paper. You cannot have these things. So, in 3-dimensional structure, when you talk about the sharing of one particular face of an octahedron, so these all 4 groups in one plane, and you have the acetate group in the other, and or the other group in below the acetate function. But the catalytic side, that means when you have the oxo-hydroxo connectivity, which is basically controlling your manganese distance as well. And this particular case, these 4 atoms basically play that most characteristic role for its corresponding reactivity with hydrogen peroxide. And sometime we see that we can have this group the carboxylate function. This carboxylate function, if it is without the substrate molecule or without the species on which it is reacting is there. So, the entire substrate, that means the carboxylate end, which is connected to manganese, can go and remain as a free carboxylate unit, because sometimes we consider that they are not bound to manganese, but they are some distant groups. So, distal histidine, distal acetate or distal glutamate unit we call, because they have some role to play for interactions which we consider as some hydrogen bonding interactions. So, these hydrogen bonding interactions are sometimes very much needed for proper positioning of the different substrate molecules. So, we need to react with hydrogen peroxides. So, we will bring hydrogen peroxide over there, and whatever reaction we are thinking of here, that can be on this diamond shaped M2O2 unit, that is sufficient for our catalytic cycle. So, one is oxo, another is the hydroxo one, and this particular manganese. So, if they are not completely fulfilled by the donor groups. So, what we have seen on the right hand side, the manganese was loosely bound to your glutamate unit, which is highly distorted. This particular side, the glutamate unit was using for the corresponding distorted coordination. So, this distorted coordination is now moved. So, you can release one such oxygen connectivity from this manganese, and your hydrogen peroxide can go and bind to this particular manganese, and this sort of binding and coordinations are well established now when we study hemocyanin and all this. So, binding of all forms of peroxide linkages starting from your oxygen, superoxide and peroxide groups to the manganese site is well known now. So, you have this particular positioning of the hydrogen peroxide. So, this is the hydrogen peroxide. So, why it is binding in this form? Why it is not going to bridge with this bind to this oxygen? Because this particular hydrogen can interact with this carboxylate unit. So, we have this oxygen of this carboxylate unit, so that means not only the manganese is responsible for the proper coordination from the hydrogen peroxide with one of the oxygen atom on it, because the leveling experiment can also establish that which particular oxygen is responsible for this binding, but this corresponding hydrogen bonding interactions with some terminal carboxy end can also go for the proper positioning of this hydrogen peroxide group, and not only these two interactions. So, this is one, the coordination, the second one is the hydrogen bonding interactions of this hydrogen with this oxygen. Also, you can have these two groups, one is OH and another is O2 minus. So, this O when bound to your manganese site, and these two manganese, this is also +3. So, manganese compared to your +2 oxidation state. So, this oxygen is again nucleophilic in nature. So, this will also show some interaction with this hydrogen. So, these are the very well known interactions, because we will see for the other example for this dimanganese compound, that whenever we have this oxo and hydroxo function, and when this hydroxo function is available and it is bound to such manganese 3,3 center, this oxygen can always have some tendency, because there is some little bit polarization is always there. So, this hydrogen can go for hydrogen bonding interactions with other carboxy end or any other group, and ultimately it can be removed from there. So, the interconversion of these groups, that means always depending upon the coordination and the manganese site, is very difficult to predict for all these cases, whether you have a bound or bridged water molecule or a hydroxide function or an oxide function, because you cannot control the corresponding pK values of the groups or the protons attached to these oxygen atoms. So, once this connectivity is established, in the third step what we get, that this particular oxo function can now take up this hydrogen. So, that interconversion what I am talking about, that interconversion of oxo and hydroxo function. So, this will become hydroxide function, and this hydrogen can also after bound. So, this hydrogen can also be released So, hydrogen peroxide is basically giving you 2 protons together. So, this one will be, it was OH, this one will be OH2, and the corresponding oxidation state of manganese will also change, and like that of your binding of myoglobin or hemoglobin molecule, this is now we can write it as a dioxygen coordination to that. So, that is the thing. So, in one case we have seen that in case of hemocyanin type of system, that 2 centers are utilized, where initially in the low oxidation state of copper, dioxygen is bound to the copper sides, then electrons are transferred to dioxygen in its antibonding orbitals. We move from dioxygen to superoxide, then to peroxide. So, now the reverse thing is happening, you have added hydrogen peroxide. So, hydrogen peroxide is now slowly moving towards a dioxygen molecule. So, that is the thing what we are getting for the catalase reactivity. So, you have the bound dioxygen molecule now. So, you put now the second molecule of hydrogen peroxide. So, when we bring second molecule of hydrogen peroxide, this O2 molecule as dioxygen is released from there, and one water molecule is also released, that means this water molecule. So, these are weakly bound now, originally it was hydroxido, it is now weakly coordinating to these 2 manganese sites. So, it will be released as water, and these 2 oxygen atoms like oxygen molecule is also released. So, what will you get at this point? That you have manganese, both the 2 manganese sites you have, and now a peculiar arrangement from this particular unit is operating, and sometimes it is attached to H also. So, this had H2O2. So, this when it is terminally connected to 3 oxygen. So, that means you have initially OAH. So, hydroperoxide is forming, then that hydroperoxide is interacting, that means this oxygen is interacting little bit with one such hydrogen atom. So, this still it is OH. So, you have manganese 2 and this is also manganese 2 plus, and from that particular unit. So, you have this free carboxy and you have to write so at this point. So, you need the elimination of water molecule now. So, 2 water molecules and 1 dioxygen from 2 molecules of hydrogen peroxide. So, this particular thing has been proposed from there, that you have OH2 is released and this will remain as oxo function. So, it is a direct oxo transfer agent. So, hydrogen peroxide is functioning as a direct oxo transfer agent for your this manganese. So, you have already you have OH. So, this OH is there and this is giving your corresponding oxo site. So, at this point the situation is such that you have the corresponding this OH2 is releasing, and simultaneously both the manganese sites are available to activate your hydrogen peroxide molecule, and this particular coordination, that means one such oxygen, that means you have this OO stretching frequency you can monitor. So, it is a symmetric bridging. Because the corresponding Raman and IR stretching frequency is very much characteristic. All the time if you just look at these values for monitoring this, whether you have bound dioxygen molecule or whether you have some hydroperoxide interaction or it has been transferred to peroxide interaction. So, symmetrical mu bridging is possible there, and we have mu bridging peroxide complex. And at this point, when it is terminally bound to both the manganese, we are activating the OO bond. So, at this point we are activating the OO bond for cleavage. So, we can generate the original catalyst, and that can again continue the cycle. So, basic idea behind that, there are different types of substrate, that peroxide groups we know that this can be formed when we talk about the dicopper system for hemocyanin. Similarly, the manganese based system. So, manganese based system based on manganese 2 and 3, that these two are immediately reacting with these manganese sites, and here we are not forming any manganese oxo species. So, in a different sorts of binding of this hydrogen peroxide can ultimately lead to the corresponding reaction for catalase activity. So, this is the one part, that means the corresponding redox reactions we are talking about. Again manganese, this dimanganese system. So, dimanganese system can function as a very good Lewis acid. So, we do not have any electron transfer catalysis. So, it is basically a non-redox dimanganese enzymes. So, they are known as non-redox dimanganese enzymes. And one such enzyme group is Arginases. Which is responsible for the hydrolysis of L-arginine. What is L-arginine? Amino acid having guanidinium group, and this is a very important molecule, because this leads us to give urea. So, in our system, in any living organisms, wherever we assimilate the protein molecules, the nitrogen assimilation, and ultimately we remove those molecules as urea. So, this is our and they always present as different deuterionic forms depending upon the pH of the medium. So, this is the corresponding L. So, this substrate molecule can go for the corresponding hydrolysis reaction. So, these arginases will be responsible for hydrolysis of L-arginine function. So, you have the cation dependent hydrolysis, and there are two competitive pathways, one by using arginase to provide L-ornithine and urea. That means, this part will go as urea. So, the guanidinium cation. So, this particular substrate is, you see that you have deuterionic form. So, these charges are there. So, binding of these within the enzyme molecule is sometimes facilitated, because if you enzyme functions have some different carboxylic ends, CO2 minus end. So, suitable positioning of these groups, the deuterions, you do not need any hydrogen bonding interactions or any other non-covalent interactions. So, the part on the right hand side will be clipped. Which is known as L-ornithine. Plus your urea. So, this particular part how we clip using this important manganese based enzyme. And another interesting molecule we will not study here, but you should know that, because these are the competitive pathways where the very interesting molecule, inorganic chemists always claim that this is our molecule, NO, nitric oxide synthase. So, if you look at the substrate, the same L-arginine, you have to take out NO from this molecule. So, NO from this molecule, when you take out, here we are getting this particular part as urea, but here this part still remains with the original molecule. This is L-citrulline, C-I-T-R-U-L-L-I-N-E plus NO. So, these two enzymes basically go for competition. Why we are talking? So, this is one pathway where you can produce urea. This is another pathway where you produce the NO molecule. So, if these two enzymes can compete for this particular substrate, you never know how much urea you will have, because we want to release this urea, because in mammals one such case, that is the arginase is the terminal enzyme in urea cycle. Of the urea cycle, and because we always need an adult human being release or excrete some 10 kg urea per year from our body. So, we basically remove this much amount of urea. So, this thing is therefore important, that proper positioning of these particular substrate on again a dimanganese type of the catalyst type of enzyme will go for this particular reactivity. And here if your hydroxo function can attack over here and it can transform to this, and their cleavage of this particular group can lead to your corresponding urea molecule. and but this particular case this simply goes. So, this particular NH2 plus, that means what is happening, this is the carbonyl function, and this carbonyl function when reacting with some ammonia molecule, say, it also gives the imine function double bond NH, and that NH instead of attacking your this particular carbon, if your nitrogen is attacked by this hydroxo function, you will get double bond NOH, the oxime function. So, that oxime, if once you can make, that oxime function can ultimately release that NO function. So, this particular the NO synthesis as that particular interesting function that it can release from there from the oxime function for this NO. So, let us see for this arginase. So, the basic active site structure only will see the arginase molecule, because once you know the arginase, you will have some good idea that how the NO synthesis can also work. So, you have 2 manganese very much similar to that of our catalase site, and we simply sometimes label these as the corresponding MnA and MnB, and now you have OH bridging, and this we already know that this immediately you can go for carboxylate is now aspartate, that residues may be different. This is similar to that catalase, this is nitrogen from histidine 126, instead of glutamate residues there, we have now aspartate. This is also another aspartate. Now, this is much more distorted compared to your catalase active site. Now, you have this doubly bridging unit, and the triply bridging motif is completed. What I was just telling you, that is a single oxygen carboxylate bridging. So, now you have single oxidate carboxylate bridging again from aspartate residue of different number, is aspartate 232. So, this triply bridging unit is there, and this oxygen is basically functioning to coordinate to this manganese. So, 1, 2, 3, 4, 5, 6. So, this manganese is close to your preferred octahedral geometry, but this manganese is not. So, you have 1, 2, 3, 4, 5. So, it is still 5 coordination. It can also be 6 coordinated and in octahedral geometry if we bring this oxygen coordinated to this manganese, but this oxygen when it is pointing towards this particular region, that means it is a different one, and this oxygen is not available for coordination to manganese A. Instead, this can show, this is close to this group basically. So, it can show some hydrogen bonding interaction with the bridging OH group. So, that gives some very important idea that we have the 2 spin coupled manganese sites, because these both the manganese, because it will not show any kind of redox reactivity. Arginase is a non-redox one. So, both the manganese 2 will be there. So, is a dimer of manganese 2 and manganese 2, and since these donor groups are mostly oxygen based, you see only you have 2 nitrogen. In catalysis also you have 2 nitrogen from the histidine residues. So, they are weak in nature. So, you have the high spin system. So, it is a 3D5 system, but there are interactions. So, we call they are spin coupled, that means interactions are there. So, it is a spin coupled dimanganese system having manganese oxidation states on one as +2, another on +2, and this spin coupled system will tell you that there are interactions through its magnetic orbitals bearing the unpaired electron density and the corresponding manganese distance, which is close or around 3.3 angstrom. So, when this oxygen is not available for coordination to manganese A, rather it is engaged in hydrogen bonding interactions with the bridging hydroxide of function. So, this particular one will be square pyramidal, close to a square pyramidal arrangement. And these interactions will see that when we see the corresponding catalytic cycle, we will find that how this particular OH group will come and will interact with this manganese. So, when it is square pyramidal and it is not a trigonal bipyramidal arrangement, because this particular face is the corresponding octahedral face, and once you have this trigonal face and one such oxygen from there. So, it is basically giving a square planar arrangement, and you have the histidine as the lone coordination site from the other side, giving the corresponding square pyramidal arrangement. So, if you have some positions like this, that immediately tells us that your substrate is your L-arginase, and binding of this substrate to the active site is very important. How close you can push the arginase molecule to these manganese sites. So, initially if some donor groups like the binding of catechol unit or any other phenol unit, the tyrosinase activity what we have seen, that you have the phenol groups, and those phenol groups are going and attaching directly to the manganese sites. But if these both the two manganese sites are surrounded by all these donor groups, we will find that positioning of this arginase would be difficult. And if we get some manganese, one particular manganese site which is square pyramidal, that means you have certain space, the vacant sites are available. So, those vacant sites are there which can be approached by your substrate molecule for corresponding reactivity. And we are talking about some two catalytic sites, one is the arginase activity and another is NOS, the nitric oxide synthase, and they both of them will compete for this particular substrate, that means the arginase. So, whether you are producing urea molecule or you are producing the nitric oxide molecule, that will be dependent on your level in our body, which can vary from one person to other, our level of this enzyme concentration. So, one is the substrate concentration, another is the enzyme concentrations. So, those enzyme concentrations for this particular arginase catalyst will tell you that we can also design something where you can little bit disturb the corresponding activity for NOS. So, that is one such mechanism we call when you have some manganese with some vacant site or vacant space or some exposed area, which are designing some inhibitors, we call So, next day when we will see this actual catalytic cycle, we will find that these inhibitors, so these inhibitors will straightaway call them as arginase inhibitors. So, inhibitors are some group which will make your catalyst inactive for your arginase substrate. So, designing for these inhibitors is not that all groups, like the very small groups, sometimes we even test the inhibition of catalyst inhibition by some small inorganic anions like azide, thiocyanate, cyanate, etc. These small groups which can have some affinity for manganese binding. So, once you put that, so they goes and bind to that manganese site, and that manganese site will not be available for any kind of reaction with your substrate, which is your arginase. So, when we put these arginase inhibitors, what will happen? It can enhance the substrate pool. That means the concentration of the available substrate. So, which is there in the central pool, that means it is available for arginase activity as well as NOS activity. So, we can enhance the substrate pool for nitric oxide biosynthesis. So, that gives us some clue that how inhibitor design sometimes can help us. That means, designing these inhibitors, we do not want to see much activity from these arginases. So, we want to increase the corresponding production of NO. These are very good neurotransmitter molecules. So, they can activate your neuron and other positions. So, they are very much effective in that way. So, how you can increase the nitric oxide biosynthesis? You reduce the corresponding activity of these arginase molecules such that your NO biosynthetic pathway will be activated and you can be able to produce more and more NO molecules. So, next day we will see the entire catalytic cycle where the binding of the substrate would be important, and we will have manganese A as well as manganese B for this binding, and both these two sites are in +2 oxidation state, and already we know that we have this particular OH function, OH groups. So, apart from this catalase where we go for the redox, now we have no liability for changing the oxidation state of manganese. So, manganese can remain as manganese 2 plus, and which is very much similar the atomic size and all these, because sometimes we can compare this as the charge and all other thing is very much close to your magnesium what is present in our photosystem. So, this hydroxide function and your that tail of the substrate, the guanidinium function. So, this tail of this function which is this NH2 plus. So, tail of these functions. So, it is bound. Now, you know what will happen, it is nothing but your attack by this bound OH function to produce the urea molecule. So, very simple strategy for that. So, nature has also devised for this simple strategy. So, first you have the binding of the substrate, because this particular part which is a charged one and also positively charged, you remember that this is the beauty of this particular enzyme, that is positively charged. You cannot consider this particular group or this unit as a ligand, otherwise it will be negatively charged, it will have the lone pair of electrons, it will not go. So, this sort of binding we will see how it is positioning and how OH is attacking your that particular end of the arginine. Thank you.